it's time for the second talk of today, and this is on Professor Asima Chatterjee, and this is by Professor Sivapriya Kirubakaran. And to proceed, we'll first request Professor Michel Danino to introduce Professor Sivapriya Kirubakaran to the audience. Michel.
So it's my great pleasure to introduce my colleague and friend, Professor Sivapriya Kirubakaran, and she is a distinguished chemist, and she has strong interest in biology also. And at the Indian Institute of Technology, Gandhinagar, where she's associate professor currently, she's also part of an important lab, which is the Cancer Chemical Biology Lab, where she's conducting a lot of research. So I think she's ideally placed to tell us about this figure, another little known figure of Indian chemistry. So Sivapriya, over to you, please.
Thank you, Professor Danino. Thank you, Sudipta. I'll just start with sharing my slide. Is it full screen?
Yes, it is full screen. Perfect.
I'm changing slide. Just check whether the slide changes. Is it changing?
Yes, it is perfect.
Okay. Thank you so much. It's been the last lecture has been really scintillating, and then knowing how scientist in physics achieved her own way. So I'm going to talk about the scientist who was in chemistry but achieved her own way to be a scientist. I'm not actually going to categorize Professor Asima Chatterjee into a chemist or a biologist or a physicist, because what her contributions through this lecture series, I also learned about her a lot. So I'm very proud to present some of her work, and also to tell you something about how we are going to contribute to what she left and where she left. This is the doodle picture that everybody would have seen. That's actually Google in the honor of her 100th birthday. Let me just check. So you see some rings, a benzene ring. This is not a benzene ring. There is so these are all part of natural products. So that is what it depicts. And that entire doodle is in green. So you can see that she is a very nature-loving person, but she worked enormously in bringing the local medicinal plants to work in the human physiology. Okay, so she is also the first women scientist who was awarded a Doctor of Science from an Indian university. So I'll just start with her brief background from where all the beginning of her career started. She's born as Asima Mukherjee in 1917 in Calcutta. And her father was a doctor, and also he had a love for botany. So that is where she developed her interest in medicine, especially on plants. And then she did her higher studies in chemistry. And then she passed from University of Calcutta, and then she joined University of Calcutta to continue her master's in specifically in organic chemistry, because organic chemistry is part of chemistry where you study more on anything that is relatively away from physical chemistry, but very close to biology. So anybody who is interested in botany and biology used to actually do organic chemistry. Even I did organic chemistry because of my interest in medicine. So as a doctoral student, she worked with the two big names, Professor P.C. Ray and Professor S.N. Bose. P.C. Ray is a chemist, as S.N. Bose is a physicist, as many of you know. This shows that the interdisciplinary science started way long back. It's now we talk about interdisciplinary science, but both the supervisors are from different background. So her doctoral thesis actually went to a Nobel laureate called Professor Todd. He was a very big name in chemistry as well as in nucleoside biology. So she was awarded doctorate in 1944. And this is the picture that is from Indian Academy of Science. Actually has a lot of historical pictures of hers, and they have a series, Women Scientists in India. So from where you can see her husband is also a professor in physical chemistry, but he is known for his soil mechanics. This is her daughter Julie. So this is her family background. So when she started doing research as a PhD student, as well as started her own career as an independent faculty, those were the trying days of research, you can say, because as you can see this figure, there's a cartoon that was actually dedicated to her from an illustrate. You have a big RB where she's actually extracting something. Okay, you see the switchboard, the light, anything that background you see, this is the entire, this is just the scenario of the laboratory condition in those days. And natural product characterization is what she has taken up at that point, and also the synthesis of those natural products that has been isolated has been extremely, extremely difficult to characterize, because for every spectral analysis, now in our campus, we have NMR, we have IR, we have mass, you name the any characterization technique, we have it. But for any spectral analysis, that has to be sent to abroad. That abroad usually means it's either Europe or it is United States. Okay, so it's not that we have to send it to the neighboring countries. So it usually takes months together to get one spectral analysis at that point of time. And for any small milling of plants, I mean, for that, her lab people have to, and she herself has to actually go to the workshop, which is there in Yadavpur University. I assume that is pretty far away from Calcutta University, so that those days. And for UV measurement, which is the simplest measurement, she has to go to Bose Institute. Okay, so you can just figure out that what was that era that she has been working. One of her I think first few students have mentioned in one of the book that they used to get a research grant of 300 rupees per annum. Okay? And then the students used to get maximum 150 rupees per annum if they are brilliant. Okay, so you can think about the era that we can't even imagine. And this is one of the first pictures of hers where you can see that Professor Chatterjee is with Professor Guha, who is a father of modern biochemistry in India, and also with other scientists, Indian scientists in Cambridge. I just wanted to flash that this is the era where you see only scientists who are men and hardly women scientists, and especially chemists. So I'm going to just focus on three major research problems that she handled beautifully, where she isolated these compounds from natural products, and then she also characterized them. And then I'm going to talk about, because I know this audience is mostly of a general, not only belong to chemistry. I'm going to keep the chemistry slides very minimum, but you please stop me if you have any questions in between. So the major natural products that she worked are on alkaloids, coumarins, and terpenoids. Okay, so they might sound Spanish or French to you. So this might sound even more difficult. Okay, so an alkaloid, I'm going to define as Wikipedia says. It's an alkaloid is a class of basic naturally occurring organic compounds. It will contain at least one nitrogen. It is what is the definition says. As you can see here, these are some of the part or the hardcore of any alkaloids that you want to study. And there is another natural product family. It's called coumarin, which has a benzene molecule and with two adjacent hydrogen atoms replaced by a lactone-like chain, forming the 6-member ring heterocycle that shares 2 carbons with the benzene ring. Okay, that's again the definition of a coumarin. And then the third class of compound that she worked is actually terpenoids. Okay, so terpenes or terpenoids. So these are all large and diverse class of naturally occurring organic chemicals, and they derived from either 5 carbon species or 6 carbon species. But this is very confusing, right? This is too much of chemistry. So let me take you some pictorial representation of what are they. So if you see alkaloids, this is what is the explanation I gave you, but they are naturally present and they are naturally very, very important for the plants for their defensive mechanism. So these are some of the very known alkaloids. Caffeine, obviously we know, quinine, morphine, nicotine. So these are all the alkaloids that is actually produced in the plant system to protect themselves. Okay, and eventually we are actually using that as our own medicine or etc. etc. Okay, so these are the, if you want to remember alkaloids, you can remember plant defensive mechanism. coumarins that we almost every day use. So dalchinis and star anise and all these kind of a bark spices have this particular scaffold called coumarins. So terpenoids is one of the favorite fruit for everybody. So this linalool, alpha-pinene, this feryl alcohol, all of them are present in strawberries. Okay, so any berries which are very beautifully colored, so have these terpenoids. They are antioxidants, they are very good, and they are actually part of it. But as I show you here in the figure, it is not so easy that you squeeze a strawberry, you are going to get all of them. No, they are actually produced in very, very microgram quantities or sometimes nanogram quantities. But why are they important? That is where that she started studying these plants. So to start with alkaloids, so she almost, Professor Chatterjee almost worked for 4 decades in only alkaloids. So I'm going to show you a couple of examples where she isolated these alkaloids, and then eventually after few decades only she could characterize them. That is because again the lack of characterization techniques that were present at those times. So this is the first alkaloid that she has been, I mean, she has been very famous for working on this Raolfia alkaloids, because as you can see here, the structure, it's quite complex. Okay, so it has 1, 2, 3, 4, 5 rings. So there are 3 6-member ring. Again, this is another 6-member ring, 4 6-member ring, and 1 5-member ring. So during her PhD, she worked on something called indole alkaloids. This is a scaffold, a basic structure of indole. That is where when she started her own career, she has been started working with this indole alkaloids. They are very important and they work as a central nervous system stimulant, and it also used as a local anesthesia. Okay, so, but though the plant applications of this plant is known, but nobody knows what is the scientific background for that. For that, what you need is you have to extract this important one of the important chemical. That's what we call the scaffold. Like they say, this indole alkaloid is the most important scaffold. So this has to be extracted. This has to be characterized. Only when you do this, then you will understand whether this particular molecule is important for this action, whether this has a stimulant action or it has a non-stimulant, is it toxic? So you will not know unless otherwise you scientifically actually study them. Okay? As you can see here quickly, all the transformation to make this particular scaffold, that is, this is not the full molecule. As you can see, this is a full molecule. So there are some portions which are actually still missing. But this is the way that you characterize a chemical molecule, okay? And the structure, whether it has 4 cyclic structure, 5 cyclic structure, all this structural characterization is done in those days in 1940s and 50s only by some reactions called dehydrogenation, degradations, and some very simple chemical transformations. Okay? So the structural illustration of these molecules were actually done in 1970s, but the isolation was done by her way back in 1954. So when the total synthesis was eventually published, okay, of this molecule, as you can see here, this is tryptamine, which is a commercially available molecule that you can get, just you can buy it, and you just couple these 2 reactants to get a molecule which has only 3 rings now, and then doing several transformations. This is after 4 steps you actually get this molecule. Okay, so which is beautifully fused rings or 5 rings, and then you do a ketalization reaction. Again, you do several transformations, sodium borohydrate reduction, and finally you get rid of this protecting group, and then you get that alcohol. So, but these transformations, you see that I have already clubbed 3-4 steps here. I have already clubbed 3 steps here to not to just overwhelm you with a lot of chemical transformation. You can see this was done after 3 decades of when she actually isolated the molecule. You know why? Because the first step itself, how will you characterize this molecule without NMR, right? The NMR is most important characterization technique for a chemist, and we need to have that when you actually build up a complex structure. So even the first step would have been very, very difficult to characterize in those days. But when she had all the characterization technique, and it was really a piece of cake for her to synthesize this, because she already studied these molecules enormously well. And that is how these molecules synthesis were published in very high quality papers. So the second part of her research was on alkaloids. As you can see here, this one, you probably see this pruniformis in everywhere. You can see this, and it's a very, very common plant. But this plant has actually called an alkaloid called sarconin. This sarconin is structurally very, very diverse, as you can see here. Okay. But this structure, again, it was only, so at that time in 1965 in her paper, what she has mentioned is there is a tertiary amine, there is an amide, okay, and there are 4 rings. This is what the characterization that at that point would have been done. And then later on, when the synthesis was done, and then the mass spectrometry, NMR, UV, all those characterization techniques were available, then the entire structure was published. But as you can see here, from this leaves, getting this molecule is actually not easy. So suppose if you take 100 grams of this dried leaves, you probably end up in getting micrograms of sarcopene. So, but that is not enough for getting any characterization technique, right? You need to have few milligrams. That is why making this molecule in the laboratory has been a challenging effort, and she has successfully made this molecule without any difficulty. So, and many other molecules that she published later part of her career, it's been exceptionally complex, and it is not at all possible that a simple chemist actually would do all this. Any time when you have this such a fusion rings, okay, anywhere, and here you see the complexity of the molecule. This coumarin, it is, you can see here also this is 4 rings are there. Here there are 4 rings are there, but this is very different molecule and this is very different molecule. I mean, structurally you can just see the complexity, and introducing the stereo centers in each and every portion of it very precisely is not possible. And this is the most important and most beautiful chemistry that you can see. That's why natural product chemistry is always green, means that it will always stay very popular in organic chemistry, because it is just building part by part. It's building like a monument, a brick by brick. So it is like the natural products which eventually comes out has specific structure, and it has also specific function towards biology. So then the other class of molecules that she has worked is coumarins. So again, most of the coumarins are from the bark of the tree. So this one, marmin, is a simple coumarin, as you can see here, but this molecule has a lot of acidic properties which were still not completely known, whether this molecule can be a medicine, the molecule can be a toxin, we don't know yet that, but just for the beauty of the molecule, and she has actually taken this molecule and synthesized this molecule as well as characterized it in 1967 itself. So this again, another plant that you see everywhere, and this molecule, this plant also has edgeworthinin as one of the most important natural product that is extracted from here. This natural product also has a very strong extract, which is very, very similar to the long oil or any oil that you get it from the barks. So because it has this coumarin. This molecule also was first extracted by her, but later on only in 1974 that she could able to get the complete synthesis and establish the structure of this molecule. So there is a journal of natural products. Okay, so this which publishes most of the natural products that is extracted, that is characterized, and usually whoever is extracting and characterizing it nowadays, you have to get when you synthesize it in the lab, you have to get the complete characterization approved by that person. So which is becoming more like a norm nowadays, because the characterization technique that is used nowadays in 2021 and 22 is very advanced, like X-ray crystallography, 3D, 4D NMRs. So it's much more advanced now. So how did she do this synthesis of this molecule? So you can see what she has used is very simple chemicals. There is nothing fancy about it, but the way that she used them to transfer from vanillin, which is again a natural product commercially available, to this particular coumarin is only in 3 steps. Okay? So similarly, this molecule is transformed into nardalbergin, which is also a molecule that is actually isolated from the bark of a tree. So the last class of molecule that she worked is terpenoids. So terpenoids has 3 different types of terpenoid. One is mono, diterpenoid, and triterpenoids. So all of these particular medicinal plants was later on after 1970s. She spent enormous time, Professor Chatterjee spent enormous time in extracting the important ingredients of these terpenoids, which is actually called terpenes. So this is one of the most important slide. As you can see here, this data, this particular plant that was collected from Assam is 1954. Okay, but that time she was not able to actually elucidate the complete structure of this particular terpene alcohol. And then eventually in 1968, okay, but the preservation of this was still was the one that was actually helpful for them. This is actually, this photograph is from the notebook, laboratory notebook. So many students who are hearing this lecture, is very important to actually maintain your lab notebook data so preserved, right? So otherwise nobody would have known this particular terpenol, and now this terpenol is actually used for several medicinal, which is already approved by Ayush, but it is still not completely commercialized into a medicine yet. But in the Ayush website, you can see that this has some medicinal property, which I will eventually tell you in the later slides. So, but you see the timeline that when the structure was elucidated, which is in 68, but when the first specimen was collected, and you see how much of energy and how much of passion that her group would have had along with her to proceed with this. Okay, again, this is another molecule that was actually characterized by Professor Chatterjee, and also the synthesis was eventually achieved. So when we tell about any characterization or a structure of a molecule, it is very important, because depends on the particular structure as well as the application of these natural products. There is not only 1 or 2 chemists who will be working on this natural product. There will be many natural product chemists who will be working on this product. Okay, that is why it is important to extract these common chemicals, which might eventually show brilliant biological activity. So these are all other structures that were actually established by her during the period of from 1950s to 1970s, which were isolated and completely characterized by spectroscopic analysis. And all of this molecule eventually were synthesized by several other chemists all over the world, and then high quality papers were published. But if she wouldn't have, and all these molecules, whatever I have shown till now has been isolated from only Indian subcontinent. It is not from anywhere else. Okay, so you can see that our country had so much of plant products, and still has a lot of plant products that has to be characterized, and which we still don't know about the medicinal properties of those plants. Okay, so again, this fruit, many of you would have seen this. This is an important aphanin. It's an important plant product, but a lot of terpenoids, which is part of this plant product, is actually still not known. This is only one of the terpenoids that is actually isolated. Okay, I will skip this. Okay, so why I brought this picture in between is, this is the time that when she was extracting, she was actually characterizing all these complex products. So the picture I showed you was in 40s, and this picture is around 70s, and you can see that in 25 years of her time in the Calcutta University, she encouraged so many women scientists, and all of them are her PhD students or master students, I believe. That is what is written in the Indian Academy of Science website. And this is Professor Linnaeus Falling, who actually visited her, looking at her brilliant work. So her major achievement or major contribution has been taken a lot of time, because it is not very, very easy to bring a natural product, which is a plant product, to a market. So the first one of them is for the treatment of epilepsy, that is actually extracted, isolated from water fern, and then which is a product called marceline, which is now given as a therapeutic application in epilepsy by Ayush. It is approved by Ayush. Another plant product that is from Brahmi. It is everybody knows it's for memory enhancement and cognitive improvement, which is also again approved by Ayush, which is there in the market. The last one is a commercialized product called Ayush 64, which is for which has anti-malarial activity. These are the ones that she literally dreamed about bringing a natural product into an application to medicine. Okay, so these are the major natural products that is actually developed in her lab, which is also now commercialized as medicines. So this was the recent picture, I think, which is the only color picture of her group. Now you see many of them are women scientists from her, graduated from her lab. So now quickly wanted to shift gear to this modern era, that how her natural product synthesis isolation has impacted and started many more plant biologists or plant chemists to come together. So there is a huge emphasis on something called chemical ecology. Okay, so where several scientists study what are the natural products that has a lot of implication on biology. As you can see at this picture, I have taken it from Berkeley. So where they use metabolic engineering is something which is very, very important for human physiology as well as in plant physiology. This metabolic engineering, if used well, okay, so what are the things that you can achieve? I'm sure that if it's mentioned or not mentioned, because of the several natural product chemists, this metabolic engineering in plants is definitely achievable. Okay. So this plant everybody would have seen. It is all over in our campus also. Okay, so in Tamil it's called Nitya Kalyani. So rather than just being a pretty plant, this also produces an anti-cancer molecule called vinblastine. Okay, so this is a tropical flower. That's why we found it in India also. It has vinblastine, but this vinblastine is obtained from this plant only in microgram quantities. So it needs 35 different enzymes, okay, just to make this vinblastine. Okay, so then you can think about the complexity of this molecule. So if you want to make vinblastine, you need these 35 enzymes in the lab, okay, which is I think it's very, very difficult, and it will become very, very expensive for me if I want to buy all these 35 enzymes, which mostly it will not be commercially available. But how do a plant synthesize an anti-cancer drug in 35 steps? And why does it synthesize? First of all, plants doesn't have cancer, but it is synthesizing a anti-cancer compound. So you know why? We don't know. But Sarah Connor can have an answer, right? Okay, but this is not the Sarah Connor I want to show you. So this is the Sarah Connor, who started as a plant chemist or biologist in MIT, but now she is in Max Planck. So what she does is, she is engineering a specific novel protein, okay, so that the protein produces what she wants, okay, in the plant. So that means that she is using a plant as her test tube or a round bottom flask. So, but engineering new genes in the plants has been already done, right? So it is not everybody knows this BT, Bangan, BT, so many things. If you want to make a pesticides within the plant, it is also possible. But what she has done is very, very novel and very new, that she is actually making this reshaping not only the natural product that she wanted, but she's also reshaping, mutating the small proteins, which synthesizes this natural products eventually. So now we have vinblastin or vincristine, both are from the same plant, which can be made in milligram quantities. Okay, and the plant makes only vinblastin. The plant doesn't make any other things. Okay, but the plant grows very healthy, very normally. As you can see here, she has a beautiful greenhouse. When I was in Boston, we visited her lab, was just flourishing that time. So it is the most beautiful lab, right? Because it has so much of flowers and everything is very green in the lab. So now she moved to Max Planck, where she heads this chemical ecology, entire chemical ecology program of Max Planck. Okay, so what she has done is that she has engineered this genes, okay, from the plants, which can be transformed into a bacteria, okay, and where you can attach chlorine and bromine to vinblastin and vincristine, or whichever molecule that you want. Okay, so this root actually produces a lot of natural products. So what she is doing is this entire, sorry for this picture is not very, very clear. This entire root that you have also seen what Professor Asima made in her lab. So these kind of indole alkaloids that you want, you can engineer the plants, you ask the plants to make it. And these roots have molecules which has a bromine and chlorine here instead of this X, or whatever you want. You want a methyl, you want a chlorine, you have a bromine. That means you are actually telling the plants that you make this specific molecule. So, which is very, very cool according to me, and very, 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 very important and very interesting. Because eventually, this is the way that the true green chemistry can work, right? What you have solvent here is water. What you are giving to this plant is a natural things, natural sunlight. And the plant is actually making your anti-cancer drug. So similarly, there is a Professor Stephanopoulos in MIT who engineers bacteria, and he makes Taxol in tons. Okay, so which is again a natural product, which is synthetically very, very difficult. It has 47 steps to make, but he makes it in one step. Okay, this particular area of Saras is very well known and is highly cited, and it's very, very inspiring to read. And another one lab I just wanted to mention here is Professor Beng's lab from MIT Whitehead Institute, where you can see here it is very, very close to the betel leaves that we have. It is not a betel leaf, but it's called a pepper plant, which is also available in many tropical countries. Mostly it is available from Hawaii. When the lab was started at MIT, Whitehead Institute, and the roof of Whitehead Institute was completely converted into a plant kingdom. And since Boston has only 4 months of sunlight probably, and these plants were actually not growing. Okay, so these plants need a lot of sunlight, and these were actually brought from Hawaii. But now what they have done is they are using these plants to make something called kava lactones, which is anti-anxiety properties it has. But this is, if you want to isolate them, they are available only in very, very microgram scale. But if you want to see, as you can see here, it has to be grown for 5 years so that the roots can accumulate maybe microgram quantities of kava lactones. But it is known in Hawaii, in many tropical countries, that the extract of these leaves and the roots can be used as an for anti-anxiety properties. Okay, so they actually drink the tea of this particular plant. Okay, so now what they are doing they are engineering particular proteins, okay, and then keeping this plant as a biofactory, okay, and making this kava lactones. But that is, it's possible because every biosynthetic pathway that leads to this kava lactone is completely studied and characterized. Now those characterization has become much more easier than what Professor Asima was doing. So now you can particularly identify the gene that makes this molecule and completely modify it to see whether you, instead of this R1, R2, R3, do you want a methyl, do you want a hydrogen, do you want a, you just feed that particular starting material. Accordingly, you will actually get the final product. So this opens up for a lot of new therapeutics that can come up, and which are actually natural products, right? So it will have its own side effects, but it will not have that the toxic effect that we see for chemically synthesized molecule, because this is mostly biologically engineered. So again, going back to Professor Ashima's contribution. So what she has contributed in this 40 years of natural product research has given her almost all the awards in India, and she has been most celebrated doyen of chemistry, as everybody says. And she is also the first elected General President of Indian Science Congress. I read that speech in the Indian Science Congress in front of the Prime Minister was scintillating and very, very important talk, that she actually saw the vision of Indian science at that time. So apart from all these contributions, scientifically and getting award, what she had in her was enormous passion to bring this natural product molecule into clinic. Okay, so that is how her entire studies has been. Beyond that, she was also an excellent human being who has taken care of everybody who was in her life. And I think this is one picture that I think her daughter Julie shared, where she was attending Kali Puja in San Francisco when she went to give a talk in San Francisco, I believe at that time. So what we have here is, if you want to continue her legendary contributions, we have the entire plant kingdom or natural kingdom here. And we also have enormous technological advancement in terms of artificial intelligence and machine learning, where you can actually do what she has done for 40 years. You can actually do it for 4 years, but the knowledge that she created has to be very importantly, we have to study how each natural product is important and each natural product can be created. So because for even technological advancement, we have to feed the data, right? Unless otherwise you know how a natural product is characterized, how natural product looks like. If a natural product structure is not known, then we cannot even do anything with the artificial intelligence or machine learning, right? So we need to characterize that. So that is the forward that a chemist can take the natural product synthesis as well as natural product application forward. So I would like to thank specifically first to Professor Asima Chatterjee. If I wouldn't have doing this lecture, I wouldn't have known many things, which is personally very, very inspiring for me. The era that she has worked is nothing compared to what I am actually working in IIT Gandhinagar. So I should be probably 500 more times hardworking than her. So hopefully I will work harder after this, after reading about her. I'm extremely thankful to IIT Gandhinagar. My funding agency recently, BIRAC, has given a grant specifically to take some of the natural products to market. And I thank Shidipta and Michelle for giving me the opportunity. And my 4 students who try to help, I mean, they not try to, but they helped me to get a lot of pictures and a lot of natural product synthesis, all those slides for me, Saumita, Harita, Delna, Panindra, and all my group members. And I'm also proud to say that our group at IIT Gandhinagar has a lot of ladies. So thank you so much, IIT Gandhinagar. Thank you so much, all of you. Any questions, I'll be happy to take.
Yeah, if there is any questions, this is the time. So, or any comment, please go ahead. So Sivapriya, maybe I can start with one question from a very, I have no idea about this field. So it is clear that this field, Professor Asima Chatterjee's contribution is also related to not only the technological or chemistry development, but also related to the product oriented, right?
Yes.
So how does this work for her? Whether her research was actually accomplished some very well-known product or something we know?
So these are the 4 products that right now is actually approved by Ayush.
Yeah, you mentioned that.
So for epilepsy, so all this work has started in 50s. Okay, so but every time you have to prove your drug is working on a human, right? Or so there's something called preclinical studies, clinical studies. Every time you go with a paper and say that you this molecule is working, but the first question will be asked is show the proof, right? So though we all know that we take turmeric, we take ginger, we take everything, but what is the active ingredient that is present there, which is acting on this particular biological system is the big area that has to be understood. So what she has established is she has understood the molecule very well. She has understood the final product also very well, right? Now we have to bridge the gap between that, how to take this molecule and study the mechanistically, why this is showing this property.
Thank you. Any other questions or comment?
Do you know from when these medicines are there, or?
So this is actually there in the Ayush website itself. It is available in even Himalaya. Looks like the Brahmi tablets. And
I said from when, like, you know,
oh,
since
from when? It's from 2000. So once, so she is the one who has established this Ayush college in Calcutta. I believe there is a research center by CSIR that is established with her efforts. So that particular center is trying very hard that most of this medicinal plants can be brought as medicines through Ayush. So from 2000 onwards, yes.
You know, that was a very nice and informative talk, and I came to know many things which I did not know. Of course, you know, I knew about Asima Chatterjee. I studied in Science College. Her daughter was also there. And, you know, I just noticed I had not correlated her timing and Bibha Chaudhury's timing are sort of similar.
Yes. Yeah, I also noticed from you.
That was a little surprising that, you know, how she was really well known, and but Bibha Chaudhury was not at all. Like, yeah, thanks a lot for the talk.
Thank you so much. I know it's also the chemistry, right? It's also the chemistry, like Professor, the chemistry in Calcutta is very famous, I believe, because in IISc, we always have this South Indians versus Bengalis having this, who will go to first.
That way, physics also, there are a lot of, but this, you know, she also was in Lady Brabourne College. I think that, you know, because I studied there, so I know she started the chemistry department there.
I mean, maybe there is an issue here that to understand the gender biases in chemistry and biology versus physics, we don't know. Maybe there is a comparative study, because in biology, traditionally, the female participation is higher. Maybe that has worked. I mean, I don't know.
Also, she had a continuity in the work, you know, like publication and students. And another thing I wanted to know, how many students did she have? That is also very important. Like in sometimes
she had a lot of students, especially PhD students and masters. But yeah, I couldn't get more details. But I mean, some of them are. I think most of them are very humble people, and they all are college teachers. Looks like many of them are college teachers. So that is, I mean, usually, right, it's very difficult to get the tree. I mean, we get more information on her than her students.
I was just wondering whether there's any possibility that these two women scientists we've heard of today, both of them Bengalis, could have known each other?
Of course.
What do you feel?
I feel that both of them are from Calcutta University, I believe, right?
Both of them are from Calcutta University, but then Bibha Chaudhury left Calcutta. So I don't know, like this shows the similar time, no, 37, 38. But afterwards, Bibha Chaudhury left Calcutta and then she went abroad, and I don't know, like it's an interesting question. So I asked Bimla Bhuti, who is another, you know, very famous professor from PRL, and she said they didn't have much overlap. So yeah, I was also thinking whether they have known each other, because so few women at that time.
One book, my colleague's wife, actually, she gave me, it is fully written in Bengali. She gave me day before yesterday, telling that you are going to give a talk. There is one page where it's only in English. You can read about her. So where the author has clearly mentioned that there were only a couple of women in the entire corridor of University of the college, okay, where we can see Asima Chatterjee working all the time in the lab. So like that, she said there will be only 3 or 4 women students in the entire campus that can actually see they can see while they're walking the corridor or something like that. So I'm sure they would have known each other.
Yeah.
There is a comment in the chat box. If I am not able to understand it properly. So many a times we read about the past scientists, there is a confusion about some information. For example, when I was reading about Viva Chaudhary, there is a lot of confusion about her name. So like that, do you face any problem and how do you tackle that? I don't know. I mean, is the question that the presence of gender discrimination in current scientific atmosphere in India?
Maybe I can answer this. See,
Please.
what happens is that I think this is correct because Bibha Chaudhury's name, I also. So when we were organizing the Bibha Chaudhury lecture, I was not sure which spelling to write. Then I stuck to the spelling which was there in PRL records, which she has signed. However, if you see her papers, then, she herself wrote many different spellings. So my feeling, from all that, it goes that I think she was not very, about all these things. She was a true scientist and her main thing was science. And then these spellings, there are papers which her name is in different spelling, but I am following the one which I saw her sign, that CHOWDH. So that confusion is there. So there is no way to tackle this other than to see her papers and what his name is written in papers, because that's in print. But because the print was confusing, I could go one step forward and see her own signature.
Is there any other comment or questions? If it is not, then we like to now conclude. We thank both the speaker, Professor Shivapati Goswami from Physical Research Laboratory and Professor Sivapriya Kirubakaran from IIT Gandhinagar for this very excellent talk. And we hope that we will continue this series and we will be. So maybe there is one question, and I think it's an important question. So I would request Shivapati and Sivapriya, whoever wants, is there still gender discrimination going on in case of science? So either Shivapati or Sivapriya, any of you can.
Okay, so, what I feel is that it is yes, but it is less, discrimination in the sense that, as I said, if you have heard that when we see, what I see is that many women are not able to realize their true potential. Like, for instance, I myself had to fight a lot to reach here. I was not sure whether I would have a career in science or not. But the thing is that it is, I thought it's my problem. I got married, I had a child. So, but what I see is that, it's not my problem. It's a problem for many people, many young people are still facing the problem. And it is like, after 25 years. So there must be something. It is at two levels. One is at the societal level and one is at the level of, bias. And the main reason, once the Jocelyn Bell Barnell, if you have heard of her, she is a very famous astrophysicist who discovered pulsar, but she did not get the Nobel Prize. And she said that we cannot put the same yardstick to judge men and women. So what happens is that, all the rules were made for men, and we have to adapt these rules, change these rules to see, how, it should be made women friendly, conducive to women. So those kind of things are there. So this is a whole lot of things. There are personal biases. There are, discriminations, like I would say there are implicit and explicit biases. There are social conditions. So all these accumulates to, at least I know in physics, to have less number of women, less number of awards, less number in leadership positions.
So Sivapriya, if you want.
Yeah, I mean, she has told it most of the thing, but I mean, I have been fortunate because I really didn't feel anything till now, but I know that in many cases it's not the same. But I would just say everybody that if you let somebody discriminate, they will discriminate you, right? Now the supporting system is so high level, and every place that you get support, and I think we should fight for it. It's 2022 now, so I think that we should not just stay back. We should not let anybody discriminate. That's it.
Thanks a lot. On this very positive note, let me conclude the program and let me thank all the speakers and also the participants. And please be in touch. We'll let you know the next semester program. Thanks a lot. Bye.